Checkpoint drugs work by releasing the brakes on a T-cell, but they help only a minority of patients, because only a handful of those brakes are drugged. So we went looking for the rest of them, right across the genome. This is Brakepoint, built with Claude Science. Those brakes matter beyond checkpoint therapy. They also throttle engineered CAR-T, so across the genome, which genes are the brakes, and which knockdowns push a human T-cell toward a stronger effector state. We started from a genome-scale CRISPR interference screen over 12,000 gene knockdowns across 2.6 million primary human CD4 T-cells from the Gladstone Institutes. 2 donors out of an intended 4. How do you find the brakes in 12,000 knockdowns? The reflex is to rank by significance, but at 2 million cells, that breaks down. Over 97% of the tested knockdowns clear the bar, so we rank by causal effect size instead. But effect size alone still points at the wrong genes. The biggest hits are the cell's own signaling machinery. So we add what a magnitude ranking leaves out: direction. Does the knockdown push toward the effector program or away? Now the machinery falls away, and the candidate brakes rise into view. And this isn't a static picture. Every one of the 11,000 that passed our testing threshold is here to explore. Search any gene, hover any point, and read its causal effect and its direction. It's the real leaderboard, live. And that's the edge. Differential expression finds correlations, not what to drug. Genetics points to a locus, rarely a direction. We measure what a knockdown actually does, and which way it pushes, then weigh it against genetics and the clinic. From that map, 5 candidates chosen using prior evidence, CBLB, CD5, DGKA, SMAD3, and U-3A each scored across 7 lines of evidence, from causal effect and direction to human genetics and clinical precedent. Our lead is CBLB. It's a natural off switch for T-cell activation, and inhibitors are already in early phase trials. Its genetics point the same way, and it lands in our break quadrant. CD5 and DGKA come next. We're honest about what 2 donors can support. CD5 and DGKA hold up in both. The other 3 ride on 1 donor. Known brakes aren't significantly enriched, but that's a 2-donor power limit, not a null result. So this stays a ranked shortlist for the full cohort, a hypothesis to test, not a finished answer. 5 candidate brakes, led by CBLB. It's a target the industry already drugs, so recovering it validates our method. Every figure regenerates from cached outputs with 1 command, checked against a real bug we caught ourselves. Explore the live map, clone the code, and help find the brakes today's drugs still miss. Built with Claude Science.